All right, welcome to part one of a new MCAT preparatory series. Today we're going to be starting with biochem, and specifically we're going to be focusing on amino acids, their structure, and classification. So there's pretty much four main learning objectives for this video, which are the general structure of amino acids, L versus D amino acid configurations, explaining zwitterionic properties of amino acids, and then classifying them based on structure. And so by the end of this video, you should have a pretty good understanding of these four main learning objectives. Moving into the content, this is the basic structure of an amino acid. So as we can see, right in the middle, you have your alpha carbon. Connected to your alpha carbon is a hydrogen and then an R group. So the R group is important because this is what gives each of the 20 amino acids their unique properties. Additionally, we have a carboxylic group, or sorry, carboxyl group. We have an amino group, which are labeled. So some important things to note about these two groups are that this hydrogen on the carboxyl group can actually be removed and it can act as an acidic group. And then another hydrogen can be added to the amino group and it can act as a base. And like I said before, one thing that you really should know for the MCAT is just these R groups, which I will talk about later. And the MCAT will not really ask you to have these, the structure of every amino acid memorized, even though it can be very helpful for solving problems. But more so, you should just be able to recognize which amino acids have similar functional groups and how substitutions among amino acids could impact, a protein's ability to function. So very simple. This is just the bare bones outline of an amino acid. Now moving into configuration and chirality, you will notice that there are two main categories, L versus D. And so as you may remember from organic chemistry, around the alpha carbon, since there are four unique groups typically, you will have chirality of your amino acid. And it is important to note that this isn't the case for glycine since its R group is just a hydrogen. And so now since you don't have four unique groups around the alpha carbon, it is considered achiral. But in general, you have two possible configurations for amino acids. One with the NH3 group on the left and the carboxyl group on the right as such. And then one that's the opposite, the carboxyl group on the left and the NH3 group on the right. And so the way I remember this is if your NH3 is on the left, that is your L conformation, and the opposite is your D. And so one important thing to remember for the MCAT is that you really just have to worry in biological systems about this L conformation. So you're not really going to see, D amino acids in the body, but L amino acids are, you know, they dominate completely. And so that's what's going to be incorporated into proteins, and that's what's really important in biological systems. And it is important to note that even, just a small difference as switching the chirality of amino acids can completely um, eliminate its function in a biological system. And so you typically won't be, drawing amino acids, or you won't have to answer questions about RS configurations or other stuff like that. But you just should know that all amino acids are L conform configurations in terms of the MCAT. Moving on, amino acids are also dipolar, which leads to them having Zwitter-ionic properties. And so what does this mean? Well, like I said earlier, it's possible that the amino group can take hydrogen from solution and therefore receive a plus charge. But it's also possible that the carboxyl group can lose its hydrogen to solution and then get a negative charge on it. And so this is important because these acidic and basic properties of amino acids can impact their overall charge in combination with the R group, which can impact protein folding. It can also impact where amino acids or specific peptides are localized in bigger macromolecules. And so, this can have pretty big effects on the system of like a cell as a whole. And so one important thing to consider when talking about these Zwitterionic properties is this rule right here. And that is if the pH of a solution is lower than the pKa, you can expect a group to be protonated. And if the pH is higher than the pKa, you can expect a group to be deprotonated. The way I sort of think about this or rationalize this idea is you can think of pKa as the pH where a group wants to get rid of its hydrogens. And so, for example, the pKa of the carboxyl group is 2. That means if you're at a pH of 7, it wanted to give it away at 2. Now you're already at 7, so it already gave away those hydrogens. It's already acidic. It's already acted as an acid. And so that's the way I sort of think about these Zwitterionic properties. And another thing to note is that a Zwitter technically is a molecule that has, like we see here, a negative charge and a positive charge. And, for amino acids, it has a net charge. A lot of them do have a net charge, end up having a net charge of 0. So these two charges will cancel out. And that's not the case for all of them, but it is the case for some of them. And so why Zwitter ions or these properties even matter in the first place is because they're highly soluble in water. So these charges allow amino acids to dissolve pretty well. They also have relatively high melting points because they're able to interact with each other using these charges, and they behave differently than neutral organic molecules. And so the way you might see this definition come up in the MCAT is if you were asked, for example, what is the predominating form of alanine at physiological pH? And so then you would answer it is the Zwitterionic form with, the amino group having a plus charge and the carboxyl group having a negative charge. All right, so moving on, we're now going to be looking at the structure of all 20 amino acids, sort of grouped by their properties. And so in studying for the MCAT, I think it's really important that you have these structures memorized and you have really a strong understanding of these structures and how these R groups and how different R groups of all these amino acids impact their physical properties or, sort of determine their physical properties. So we're going to be going through all 20 amino acids. I would definitely recommend, pulling out a piece of paper and drawing out the names of the amino acids, their 3-letter codes, their 1-letter codes, and their structures as I go through it as well. Okay, so without much talking, let's just get right into it. So we're going to be starting with the nonpolar amino acids. So these are nonpolar. They're going to be pretty much thriving in hydrophobic environments. And so these are important because they really do have a strong impact on protein folding and because of their need to be in a hydrophobic area. So they don't like to be exposed to solution. So moving on to structures, first we're going to start with glycine. It has a 3-letter code GLY and the 1-letter code G. Its R group is simply hydrogen, which I sort of talked about earlier when we were talking about the L and D configurations. Super simple, super easy to memorize, and not much to know about it. Moving on, we're going to go to alanine, 3-letter code ALA and 1-letter code A. And its R group is just a CH3. Then you have valine, which has the 3-letter code VAL, the 1-letter code V, and it sort of looks to me like an upside down V, which is how I memorized it. So you're going to have a CH group connected to 2 methyl CH3 groups. Then moving on, you have leucine, which is very similar to valine, except it has an additional CH2. And then it's just the structure of valine. So, if you know valine, you know leucine because you just need to add one more CH2. And leucine has the 3-letter code of LEU and the 1-letter code of L. Then you have isoleucine, which has the 3-letter code ILE and the 1-letter code of just I. And so this one has a CH group connected on one side to a methyl CH3 and on the other to a CH2 that is also connected to a CH3 methyl. Then you have methionine, which has the 3-letter code MET and the 1-letter code M. And methionine is, besides for all the ones we've seen so far, which are just hydrocarbons, methionine is unique because it does have a sulfur. So it's going to have two CH2s, but sulfur, and then a CH3. So unique because it has that sulfur right there on the third group after the alpha carbon. Then we're going to be moving on to proline, which again is unique because it sort of forms a ring using its R group with the amino group, with its own amino group. And so one thing to notice about proline is that it does have an NH2 non-NH3, and that is because of this connection to its own R group. And so there it is. That's really, these 7 amino acids are your nonpolar amino acids. And, what's important to know about these ones is just that they like to be in hydrophobic environments. They don't like to be, near solution or accessible or have solution accessible to them. All right. So moving on, now we're going to have our aromatic amino acids, which these ones are typically, or they are bigger. They have, aromatic rings. And so what's unique about these three, and specifically tryptophan, is that its aromatic ring can be used for structural studies using light because it does absorb and emit light at, different wavelengths. And so scientists have been able to utilize this feature of tryptophan to study the structure of proteins, which is pretty unique. And it is a fact that, might come up, in your studies or maybe even on the MCAT. So moving on, we're going to start with phenylalanine, which has the 3-letter code PHE and the 1-letter code F. And so unlike all the other ones, which, their 3-letter and 1-letter codes are pretty straightforward, this one, you just remember, has an F. And so the structure of phenylalanine is a CH2 group followed by a benzene ring, which I'm not going to draw out all the carbons, but, it is just a benzene ring. Okay, then you have tyrosine, which is TYR for 3-letter code, Y for 1-letter code, and pretty much the same exact structure as phenylalanine, except it has an OH group here. And so the reason that tyrosine has that red star above it is because this is the first time we're seeing an amino acid that has a group that can be protonated. So this OH can be protonated, like I've drawn here. It can be deprotonated and have a negative charge. And so it's not really important to have these pKa's, of this protonation event memorized, but it is very helpful to have a general idea of, what pKa this specific group has. Because, it might be that you are asked, okay, so at a pH of 7, what's going to be the charge of your tyrosine? And so then you need to know that the pK of tyrosine is around 10.5. And so this group is, not going to be deprotonated. It's still going to have that H attached at a pH of 7. Okay, and lastly, among the aromatic amino acids, we're going to have tryptophan. Now, this group can be a little bit confusing to draw, but I'll try my best. So first, we're going to have a CH2 bound to another carbon, and this carbon is going to be bound like this to a nitrogen. Sorry, let me just fix that up. So there is another, carbon here, double bound to a nitrogen that is bound to a hydrogen, and then just a benzene ring down here at the bottom. And so, like I said before, tryptophan, is one of those amino acids that does have a specific wavelength where it's excitable. It does have, specific emission wavelength and absorption wavelength. So that's one of the reasons it's been used to study structure of proteins in the past. All right, so now we're moving on to our polar uncharged amino acids. And what's unique about these 5 is that they're going to be able to make hydrogen bonds. They are polar, so they're going to be in hydrophilic environments. They are going to want to be able to access solution. And so now let's just move on to the structure. Serine, 3-letter code SER, 1-letter code S, as a CH2 bound to an OH. Threonine with the 3-letter code THR, 1-letter code T is very similar. It has a carbon bound to a hydrogen, a CH3, and an OH. So you see very similar between the two. Obviously you can see where the polarity is coming from in both of them, and these lead to its hydrophilic properties. Then you have asparagine, which is a little bit unique. It's a CH2 bound to a carbon, double bonded to an oxygen, and then an NH2, and it has a three-letter code ASN, one-letter code N. Very similar to glutamine, which is three-letter code GLN, one-letter code Q. Not sure why it's a Q, but it is. Very similar to asparagine, just one more CH2 group. And there we go. All right, now we have cysteine, which is a little bit different because it does have sulfur. So it is the only amino acid, besides from methionine, that does have that sulfur. And so it can participate in, sulfur disulfide bridges, which is something we'll talk about in a later video. But another thing to note about cysteine is it does have a pKa around 8.4, so around 8. So at physiological pH, it will be protonated, but once you start going above 8, it can be deprotonated. Okay, so now we have two acidic, negatively charged amino acids. So both these amino acids at physiological pH will be negatively charged, um, and that's really what's unique about them. They are very acidic. So you have aspartate with a three-letter code ASP, one-letter code D, and glutamate with three-letter code GLU and one-letter code E. And so similar to asparagine and glutamine, aspartate is a CH2 with a carboxylic acid, and glutamine, same thing as glutamate, just has two CH2 groups instead of one. And there it is. So very similar, as you can see, to asparagine and glutamine, but now it's just aspartate glutamate because they have that carboxylic acid group. And like I said, these are acidic amino acids, so aspartate has a pK around 3.9, and glutamate, as you would expect because of its pretty much identical structure, has a pK that's very similar, around 4.1. Now, finally, we're moving on to the basic or the positively charged amino acids at physiological pH. So first we have lysine, lysine, which has a three-letter code LYS, the one-letter code K, and it has four CH2 groups. Or sorry, four CH2s connected to an NH3, which is positively charged. Again, at physiological pH, you will see an NH3 because it has a pK around 10.5. Next, we have arginine, which is three CH2s bound to a nitrogen, bound to a carbon, double bound to a nitrogen, which one of these hydrogens is the hydrogen that's either protonated or deprotonated, and then that is all bound to an NH2. And so, in lysine, this is the hydrogen that gets protonated or deprotonated, and arginine, it's this one. And arginine actually has a much higher pKa, well, not much higher, but it is higher than lysine, and that comes in around 12. So it's pretty uncommon, I'd say, to see the arginine deprotonated. And then finally, we have histidine, which can be a little confusing to draw, but the way I think about it is a CH2 bound to a another C, which has a double bond, and you're going to have two nitrogens. So it's sort of identical up to here, except for the fact that this nitrogen up top is going to have an H bound to it, and this nitrogen in the bottom is going to have a N plus and an H. So this hydrogen down here is the one that's, can either be protonated or deprotonated. And then you're also going to have a double bond here. So just like that, that's histidine. And what's unique about histidine is even though it is basic, the pK is around 6. So this is an amino acid that, it's common for it to be deprotonated, it's common for it to be protonated, and since its pK is right around physiological pH, This is an event that can happen and switch frequently in certain environments. And that's it. Those are all 20 amino acids with the 3-letter codes, the 1-letter codes. And I hope from this video you were able to take away, at least from the structures, that structure has a huge, huge role and impact on function. And if you can understand the structure and the function of all the R groups of all the amino acids, you'll have a very easy time answering MCAT questions related to this topic.